Deoxyribonucleic acid is the fundamental software of biological life, but for decades, our editing tools remained blunt instruments, resembling axes rather than scalpels. Standard CRISPR-Cas9 creates double-strand breaks, which often trigger chaotic cellular repair mechanisms, leading to unintended insertions or deletions called indels. Prime editing introduces a search and replace system that avoids double-strand breaks, offering a level of precision previously thought impossible in living cells. The prime editor is a massive molecular machine created by fusing a modified Cas9 nickase to a reverse transcriptase enzyme. Unlike the original Cas9, this nickase version is engineered to cut only one strand of the deoxyribonucleic acid helix. The reverse transcriptase component is the engine of change, capable of writing new genetic information directly from a ribonucleic acid template into deoxyribonucleic acid. This process is directed by a prime-editing guide ribonucleic acid, a multifunctional molecule that is significantly larger and more complex than standard guide RNAs. The first section is the spacer sequence, which identifies the exact genomic address where the edit must take place through base pairing.
The second section is the primer binding site, which anchors the guide to the nicked strand of the target deoxyribonucleic acid. Finally, the reverse transcription template contains the actual desired sequence change, whether it is a single base swap or a small insertion. The process begins as the prime editor complex scans the genome for a specific sequence called the protospacer adjacent motif. Once positioned, the Cas9 nickase cuts the target strand, creating a free end that can be used for the next chemical step. The primer binding site of the guide ribonucleic acid then finds the newly freed deoxyribonucleic acid end and binds to it securely. The reverse transcriptase then uses the guide as a blueprint to extend the deoxyribonucleic acid strand with the new corrected sequence. This creates two overlapping flaps of deoxyribonucleic acid, one containing the original unedited sequence and the other containing the newly synthesized edit. Cellular enzymes naturally favor the removal of the old unedited flap, allowing the new sequence to integrate into the double helix structure. DNA ligase then seals the backbone, creating a heteroduplex where one strand is edited and the other remains in its original state. The cell now detects a mismatch between the two strands and must decide which sequence to keep as the permanent record. To ensure the edit is preserved, we can program a second nick on the unedited strand, tricking the cell into using the edited strand as the template. Once both strands are repaired, the edit is permanent and will be passed down to every future daughter cell through natural division. Prime editing can perform all 12 possible base-to-base transitions and transversions, correcting the vast majority of known pathogenic mutations. Beyond simple swaps, this technology allows for the insertion of new genetic material with surgical precision without disrupting surrounding genes. It can also delete specific harmful sequences or redundant code, offering a way to streamline the human genome for better performance. Because it requires 3 independent DNA binding events to succeed, prime editing is far less likely to cause off-target effects compared to older methods. By avoiding double-strand breaks, we significantly reduce the risk of large-scale genomic rearrangements and cellular toxicity, which can lead to cancer.
One primary clinical target is sickle cell anemia, where a single base mutation in the hemoglobin gene can be corrected back to health. Tay-Sachs disease, caused by specific insertions or deletions, is another candidate for complete eradication using the prime editing search and replace function. For cystic fibrosis, prime editing can restore the function of the chloride channel protein by precisely fixing various mutations across the large gene sequence. To reach cells in a living body, we can package prime editing instructions into lipid nanoparticles that bypass the immune system and enter cells directly. Alternatively, engineered viral vectors can be used to transport the genetic code for the prime editor into specific organs, like the liver or brain. The next challenge is scaling this technology to edit trillions of cells simultaneously, which is required for systemic genetic rejuvenation therapies.
Prime editing offers a safer path to germline modification, potentially allowing parents to remove hereditary diseases from their entire family lineage forever. Beyond repair, we can use prime editing to introduce beneficial variants that provide resistance to infection, lower cholesterol, or increase bone density.
By targeting the genetic drivers of aging, such as cellular senescence and metabolic decline, we may soon program the body for indefinite health. Cognitive enhancement may also be possible by fine-tuning the expression of genes involved in synaptic plasticity and memory formation. We could rewrite our metabolic pathways to process nutrients more efficiently, or even synthesize vitamins that the human body cannot currently produce. For long-duration space travel, prime editing could introduce radiation resistance genes borrowed from organisms like tardigrades or Deinococcus radiodurans. The principles of prime editing are now being adapted to edit the epigenome, allowing us to turn genes on and off without changing the sequence. As the cost of synthesis and delivery drops, precise genomic design will move from specialized labs to the broader transhumanist community. National borders will struggle to contain the genomic revolution, leading to a global landscape where genetic self-determination becomes a fundamental human right. With such power comes the responsibility to ensure equitable access to these technologies, preventing a genetic divide between different segments of society. We are transitioning from a species shaped by random mutation and natural selection to one shaped by conscious design and technological intent.
The final stage of transhumanism involves the total integration of biological and synthetic systems, where the genome is just one layer of our architecture. The era of biological chance is ending, and the era of precision genetic engineering is just beginning, driven by the search and replace mechanics of prime editing. By mastering the ability to rewrite our own code with absolute accuracy, we secure the future of our species, among the stars and across the ages.